after you develop your hypothesis, then you can proceed to ask yourself, what are the ways that you are going to recruit your participant or to collect data and analyze? That is what we call the study designs. That is data. What is the ways? What is the method you are going to recruit your participant? Take data from them so that we have data to be analyzed. And that's what we call the study designs. And most often, we have what we call the observational and the experimental study design. So most of the study design that they fall within these two broad categories. When we talk about the observational, we are talking about either it's a descriptive or we are talking about analytical. In descriptive, we are talking about case report, case series, analytical cohorts. So in the subsequent slides, I'm going to take you through some of them so that you have overview of what you are stating in your research. Or experimental, also called trials. Most often, the three main trials or experimental study that we have, the community trials, field trials, clinical trials. So these are also three different terms. I'm sure we've been misusing them over and over again. So what is this study design about? Let's say you formulated a hypothesis, you want to choose one of the study design. Which one do you go for? I don't know if you are with me. Are we active? I want to have a response. If you are with me, type 1 in the chat. Yes. Okay, so far we are all on the same level. Good. Yes, that's nice. So good. We are following. Yes. So basically the main study design is observational and experimental. So let's look at just a quick one. When we talk about case reports, that one we are interested in single case. Let's say someone comes to your facility. There's unexpected association or event. A typical example is we have anatomical structure. Already, we all know how an anatomy is. Then there's a variation, you may want to report it. Or there's a typical example is during the COVID, they realize that there's a lot of what a particular person that is presenting with a distinct future, which is unknown, unexpected. So you see a lot of what case reports. So in case report, we are interested in one person presenting with what a single hot future, which is really unexpected. A typical example is first confirmed case of COVID-19 infection in India. Case report. So the person report that yes, we've confirmed there is what confirmed COVID-19 in the case in India. Then we have to what do a case report on it. So if there's what a single person, one person that is presented with an unexpected what result, like COVID-19 retinopathy. So they find out that one person was having retinopathy as a result of what COVID-19 person. So it means we have to report this and publish in the scientific audience that those that are saying that COVID-19 is not linked with retinopathy, then you report it. A typical example is when the vaccines, people say that it can use to retard the span. So if you find a case like that, even if it's one person, it means that as far as it's a single case, we try and do what? Report it or publish it as a case report. Then the next one is, what if there's a number of people? Then we call it a case series. So in case series, what happens is that we have a group, 5, 10 people presenting with the same or symptoms. To be example is how they discovered on HIV. So they realized that there was a group of people that are presenting with what? A particular or trends of symptoms of which is unexpected. In that case, we have to what? Investigate them, then report on them. So we can do case series. So in a situation whereby your study participant has a group, more than one, it becomes a case series. So the correct study design you're going to use is a case series. Then we also have cohort study. What is cohort? Most often, cohort is when we have exposed population and outcome. So for example, let's say we have a population. Population are exposed to what? Smoke. So it can be that this population will later develop what? A disease or it's not. What are we? It's possible that those that were exposed to what? Smoke will develop what? The disease or will not develop the disease. Or even those that are not what? Exposed to the smoke will develop or what? Will not develop. What are the various types of cohort study? We can have what we call the prospective cohort, we can have what we call anticipatory cohort and retrospective cohort. What are they? So for example, you as the researcher, you are here. Then you want to expose a population to a particular hot group like smoking. You want to what? A lot of people to start smoking. Then later on, you want to see whether they develop what? Lung cancer. In that case, you recruiting the population and giving them a particular kind of intervention or treatment and following up, then become a prospective cohort study. But in a situation whereby the exposure has come, a typical example is in what is happening in Ghana, the Galamsey style. So it has been happening that people are exposed to mercury. So you can go to that site, you recruit those that have been exposed. Then you later or trade them whether they develop a particular diseases. So in that case, they've been exposed to the what? That particular what? Either intervention or any harm. Then you are now going to start your study. You put them, then you follow them up. So it's called anticipatory. But what is retrospective? In that case, everything has already happened. So for example, you've realized that way back, people have smoked and developed what? Some particular disease. Then you are now come to start your study. So in that case, you go back to see whether these people, they what? Exposed to a particular what? Situation, and you further what? Look at it to see whether what? They develop the disease or not. In that case, in a situation where both the exposure and the disease has occurred, we call those kind of study retrospective or cohort study. Or in most publications, you only see retrospective or study. So when you are moving forward, like you are now recruiting your study population, you are adding them to treatment, then you follow up to see what will happen, then we call it a prospective cohort study. But if you are going back, everything has already occurred. You want to see what is associated with this condition. Then we look at what we call the retrospective cohort study. So a typical example is, so let's look at this case. Let's look at this. So way back in the 1920s, this is what happened. There were a group of people that were exposed to analyte dyes. Okay. Then around the 1950s, it was shown that some died due to what? Bladder tumors. Then a researcher comes to start a study to see what has happened between the 1920 and 1950. So the person is now starting the study in 2020. What do you think is the kind of study design? So let's see the answers in the chat. Retrospective, yes, retrospective. So any more answer? Retro. Good. So it means that we are really following, we are on point. Yes, so it means that since the researcher is now starting, that's good. It's good. So we are really following. So it means that since the researcher is now coming to what? Start. Both the exposure and the event or the outcome has occurred. So it means that we go back to get the data, then we also pick the outcome, then we just do a statistical analysis, see what has occurred. So this one also have the strength and weakness. I'm not going to touch on that. If you have the PPT, you can go through them. Another case typical is the case control study. What is the case control study? In the case control study, we have two groups. what are the groups? We have what we call the controls, and we have what we call what? The cases. So what are the cases? Cases, for example, they are those that have the disease. So the definition of case control is basically means that there's one group that has the disease group, and there's another group that are healthy, they don't have the diseases. So for example, people try to do a study, for example, they put only lung cancer patients and they want to find a comparison between those two groups. Those are not case control studies. Those ones, we call them a comparative or study. But in a case control study, we have a disease and a healthy group. And you have to also understand that a case control study is the typical example of a retrospective cohort study. In that case, the disease has occurred. So we go back to see what was the cause. Were they exposed to any what kind of thing that will result in what? The disease. So when you are doing a study that involves two groups, a case group and a control groups, then we call it what? A case control study. Then also have the strength, the weakness, and a typical one is also the cross-sectional study. So let's say someone, you put a population, then you put them once, then you took data from them, analyze, then we call it what? A cross-sectional. You are just taking a snapshot of the population once within 3 months, short period of time. Then we call it a cross-sectional study. Either you are going to compare them into group 1, group 2, group 3, any group. So probably it's just within a particular time, forward, short, then we call it a cross-sectional studies. And the last one is the control trials. Most often we can have, as we said, the community trials, field trials, and clinical trials. What is the difference? Community trials, mostly we are looking at some social intervention. Maybe you want to cause some life change, you want the community to shift to a particular lifestyle to see what will happen. So it focuses on a group level. That's community trials, although it's a preventive interventions. But between field trials and clinical trials, you have to understand that in field trials, we are more interested in preventive interventions. For example, is vaccines. Vaccines is going to prevent a disease. But clinical trials is more of what? Therapeutic what? Intervention. For example, chemotherapy has been developed. We want to see how it's able to treat what? A particular what? Diseases. So it's just the intervention or the process that differentiates what the trials. Other than that, they are all trials. So most often what happens is that we have example is we have the sample population. Okay. Then you administer what? Preventing intervention, or those another group with what? No prevention intervention or placebo. Then you follow them up. So clinical trials is the highest level of research. So you can follow people even up for years, 20 years, 30 years, then you still be doing your research. So at the end, you've exposed them to a particular intervention, or sometimes it can take months, 1 year, depending on the trial. Then you see whether what? It remain what? Free from disease, remain free from disease, or still they have the disease. Then you will to see the outcome. So those kind of studies, we call them trials. And this is a typical example of what? So what kind of trial is this? This one is involved with preventive intervention. A typical example is what? Vaccine. So is it a community trial, field trials, or clinical trials? Let's see. Yes, so we have, yes, so because of time, I'm going to proceed. So this is a typical example of a field trial. So we want to prevent intervention. So we have a lot of phases that we go through. And so in summary, we are saying that what you are looking for tells you what you have to do. Whether you want to go by random control trial, non-random control trial, descriptive study, cohort study, and case control or analytical cross-sectional. So when you get the PPT, you can go through this. Then we also have a lot of sampling techniques. how you are going to recruit the study population, whether you are going to recruit them through what? Convenient sampling techniques, or based on a particular purpose, or you are going to what? Do a referral. So for example, if someone wants to do a study that have a sensitive group, like the minority group, in that case, we just pick one person, then the person will also go and bring another person, then person will go and bring another person. So those kind of sensitive research, we will use the referral or snowball. We have a lot of them, whether it is simple random study that every participant has what? Equal chance of being selected. That case, the kind of sampling technique is the simple random study. So there are a lot of populations that each one will have equal chance of being selected. Or is it a systematic design we are going to use where, for example, every third person in the study population will be recruited? You have to also define how you are going to sample your population. Or is it a stratum? Already we have communities. Then based on the population, for example, this community one, stratum 1 has a small population compared to what? Stratum 3. It means that we recruit more people from what? This community than this one, because this is having what? A lot of population. So that's called proportional stratified random sampling techniques. Or we can also look at disproportional. That one, based on the researcher, you can choose to what? Sample more from a particular community or not. Or cluster, you first divide them into clusters, then you can sample them. So this are a lot of sampling technique that we can also use, but we can also do a lot of complex sampling. Like you can add that. So for example, we can first do what we call a multi-stage sampling, where we first have levels. So for example, you want to do a study in Uganda and Ghana. So first you take Uganda and Ghana, then in Ghana you adopt a simple random sampling technique. So different sampling techniques apart from the first one. So it's more of stage 1, stage 2, it's stages and how you are going to apply the sampling techniques. Then you'll be able to go. And I want to also let you know that you cannot proceed with your sampling in research without calculating your sample size. Most of these things in our subsequent webinars will be going more into details. What is a sample size? We've talked about study design, but I want to let you know that the kind of study design that you use is proportional to the kind of sample size that you are going to calculate or use to calculate it. So a typical example is when we talk about cross-sectionals, the most common one we use is the Cochrane formula, and we also use the Yamane formula. But for the case control studies, we also have a lot of them, Nesses, the Fresh, a lot of them. So that's why we have to know all these ones, because the kind of even the study design will inform you what you want to do in this next research process. Then you cannot take your data, either you are taking primary data, you are doing a lab investigation, or you are using a questionnaire. Then you cannot pick your data. Then we cannot count for a statistical analysis, where statistical analysis, we look at either the hypothesis that you are testing. From the alternate hypothesis, we look at your study design, and we look at your research objectives. This will help us to do the analysis that we are interested. Either you follow all this process, then we can adapt. So most of our webinars, we are also going to train you in how to use some of these statistical softwares. Either you are using Excel Stat, SPSS, GraphPad, and R. So all these three, Excel, we don't really do analysis in Excel again. So most of our webinars, we are going to teach you R language or SPSS. So most of it is R language, SPSS, or GraphPad. Then you'll be able to do it. But when you are done this stage, you should be able to generalize and interpret your research. So in this case, you are going to the end of your research methods, your research process. You are almost on ending. So you generalize it to the population. But what you have to take notice that is your research that you've done, is it valid? When I talk about validity, does the study population that you've recruited, is it a representation of what? The study population? Or the result that you find, can we generalize it to the other population? For example, you find out that, oh, let's say a particular high body temperature is associated with COVID infection. Can we tell or generalize this to the population? That's called the external validity. So anything that you're going to do, make sure you take into account the internal validity and the external validity. And we have to also look at accuracy. You have to make sure that we are always precise and valid. Precise means they are closer to each other. Valid means is the true value that we get. There's no falsification of results. It's very, very important. And again, when you are doing your research, we almost get to the end. Throughout your research, then you have to look at, make sure that there's justice. No group carry more than harm. You can't recruit someone and cause the person to have a disease at the end. We don't do that in research. For example, you've gone to a particular community because they have there's high level of poverty, there's disease, or there's lack of education. We are using that one to take advantage of them. You don't do that in research. Or you have to make sure that there's benefits. Benefits should be more than the harm. And again, there should be participants should be free. You can't just go and force someone to participate in your study. It should be free to participate and also to fall out. These are called the researchers. So you have to have the key point to remember that uniformity of assurance. The most one is you have to be able to train your research groups. So any research that you're supposed to do, and today, I'm very happy that most of you have joined us for today's training, and at least you've learned some things about research. And what I want to tell you that if you form a research group, you have to be able to train them on the kind of research you're going to do, who is responsible for which task, in order to produce a good research. So most of others are references, which we can further read. And I'll tell you that if you are still not following us, make sure you follow us on LinkedIn, Facebook, X, or email us for any other details. We are happy for you joining us, and thank you.
Thank you very much, Mr. Ebenezer Senu, for this wonderful presentation. You have shared tons of information that I believe will help us in our research journey. Please, the floor is open shortly for questions. If you have any questions, you can raise your hand and you unmute yourself and talk, or you can just send that in the chat box so that we respond to you. We'll do this within a period of 10 minutes. So let's be fast.
So let me see. Yes, someone say, can we get recording? Yes, we share the recordings with you. Okay, I think someone's hand is up. Yes, so let's see. Yeah, Musa, yes, you can go ahead.
Okay, thank you very much. You mentioned that sometimes you do a research where you could be the first person or among the few people who have done it. And in that case, getting, let's say, paper to back up becomes difficult. So you base on theories. So I want to know how you get those theories to back up your work. Thank you.
Yeah, so I don't know, we can set any example. I'm sure already there will be book knowledge. Let's say example, let's give example of something. Let's say you want to see that heat will make someone fall ill, okay? Now, so far as there's no study on heat relating to someone having a particular disease, what you do is that you look at the principles behind the heat. What is the mechanism of heat? Then what is the mechanism or pathogenesis of someone having a disease? Then from those kind of established related theories, you're able to link them in your discussion. because there's no really a strong established fact that already heat is leading to a particular disease. It means you are not starting. So you look at the pathogenesis, already science behind those kind of words, a scientific term. Then you can use it to establish your point. Then that will inform. So in this case, you know that in our setting, we do more of observational studies. In that case, we are relying on data. But out there, a lot of people are doing a lot of model work. So for example, most strike science studies, we do them in mouse models. So you can pick a mouse, then you kind of program it, then look at the end results. Then you can establish it. So although you've established that, oh, heat is associated with these diseases, because there's no study, it will cause for more studies that, oh, some also study and look at what is the pathogenesis, what is in the heat is causing this disease. That will also come and look at the pathways. It's the same thing like type 2 diabetes scheme. Someone find out, oh, there will be diabetes. Then someone will come and find out, oh, this particular endocrine is resulting in the diabetes. Someone also come and find other pathways like that. Then it will lead to a broader scientific contributions. I don't know if we are on the same level.
Yes, so I got you right. So you have to base on the principle,
yes,
the principle that can lead to that. You say it instead of using a certain article.
Exactly.
Yeah, okay.
Yes, because there's no already a particular article that has the same result that you have. So you have to be based on that. And people also come and be building on what you've done. We say I found this. Then they also come and be building on it. At the end, you by the time realize the whole field is out there. COVID started a lot of research. They find out they research in other fields, hematology, microbiology, every field is researching to see what is happening in COVID infection. Because someone has reported a case report or a case series of on COVID, then the molecular genomics, they will come and sequence it to see what is involving. So it means that for the first time, you say there's no data, so you're not going to publish. That's why we talk about case report, case series, then like that. But if you're doing observational studies, means you are also going to depend on principles and already established science.
Okay, thank you.
Welcome. Yes, do we have listener? Yeah, someone is saying that what is the difference between systematic? Systematic means that it follows a methodology approach. Like, for example, if you're even doing the lab aspect, you've added this reagent to this reagent to this reagent. It's kind of follow what? A chronological order. Someone can look at and follow the steps. But the step that you follow, can someone reproduce it somewhere? Can someone also do that particular methods and find similar or different results? That's what we call the replicable or reproducibility. So I don't know whether we are all on the same level. Okay, Ba is further asking whether we certificates. No, for this particular webness, there will not be certificates. But as I said, this is not our first or this is not our last webness. So the webness that will have with certificate, we will specify them that, oh, this webness, you'll be issued certificate. But this particular one is just for an information session for you to join us to see what we do and appreciate that so that we can continue be having you in our subsequent webness and also on our membership list. So there won't be any certificates. I don't know whether we have more questions. So I'm sure Solomon, I can stop sharing my screen.
Yes, please, you can stop sharing.